And this is the last time. And I'm not going to come back. So, if you have any questions, please make sure to ask me today. And as I always say, if you have any question, please stop me anytime. If you don't understand something, other people don't understand either. So please do us a favor by interrupting me. Okay, so today's topic is repair and regeneration in nervous system. It's the chapter 25 of your textbook. So let's think about regeneration for a minute. The ability to regenerate part of the body depends largely on species and the type of tissue. For example, if you cut the tail of a lizard, it will grow back. Do you know that? Yes, it does. However, if you cut your finger, it's not going to grow back. Well, you can cut your fingernail or your hair, and the cells in your gut are constantly replaced every day throughout your life. But when it comes to the nervous system, regeneration is not something you would regularly see. In fact, there are only a handful of examples where you can see new neurons are generated in the adult brain. One of such examples is mouse olfactory bulb. The part of the brain which processes sense of smell. In mouse olfactory bulb, neurons are constantly replaced in adulthood. New neurons are generated in this place called subventricular zone, SVZ, which is far away from our olfactory bulb. So the new neurons will migrate long distance to reach olfactory bulb through this pathway called rostral migratory stream, RMS. Another example is a songbird. The part of the brain which is important for them to learn and produce the bird song, called HPC, produces new neurons in adulthood. Okay, the third example I would like to show you is also mouse hippocampus. Hippocampus is very important for memory formation. In the region called dentate gyrus, inside the hippocampus, neurons are constantly replaced in adulthood. New neurons are generated in this area called subgranular zone inside the dentate gyrus. And adult-born neurons take much longer time to get mature as compared to neurons generated during development. For example, if you look at the neurons in a neonate, it will take about 2 weeks for them to make connection. However, adult-born neurons take about actually more than 2 months to get incorporated in the pre-existing neuronal socket. But all these 3 cases we looked at, neurogenesis plays a critical role in the normal function of these parts of the brain. For example, if this mouse is under stress, it will release a lot of stress hormones, such as glucocorticoids. If you are exposed to stress hormone chronically, it's bad news for your brain. What happens is the neurogenesis in dentate gyrus is impaired. And those animals would perform poorly in the hippocampal dependent memory test. contrary, if the mouse is kept in an enriched environment with a lot of toys to play with, or if it goes through a lot of exercise, like treadmill, and then you would see the neurogenesis in dentate gyrus is boosted. And those animals perform very well in the memory test. So there is a strong correlation between the neurogenesis, the amount of neurogenesis in dentate gyrus, and the performance, the hippocampal function. But this is not really a general picture of what's going on in adult brain. In fact, adult neurogenesis is very uncommon. What about human? Well, there is an interesting study using a postmortem human brain samples. Well, during and right after World War II, there was a rush of atomic bomb testing. And as a result, there was a naturally high level of radiation going on in the atmosphere all over the world until the treaty was made in 1963 to ban this nuclear testing. And since then, the radiation level gradually went back to normal. Now, if you look at the dividing cell, they have to synthesize new DNA. And sometimes they incorporate radioisotopes, such as C14. So, if there is a transient increase in a C14 level, and then you can sort of see when those cells are generated by looking at the C14 level in the cell. So, when they did that, when they examined the brain sample from the individual who was born in 1968, which is right at the peak of this radiation increase, both neurons and non-neuronal cell types in the brain contain unusually high level of C14. And they looked at different parts of the brain, prefrontal, or temporal, and all over the brain. It contained high level of C14. On the contrary, if they looked at brain sample from the individual who was born in 1952, which is before the increase of this radiation, and both neurons and non-neuronal cell types contain just normal level of C14. They looked at different parts of the brains. Those brain areas are highlighted here. None of the regions showed abnormal level. So what this study tells you is that once those cells are generated, they are not really replaced. If there is a significant amount of turnover going on, and then you should see some increase even from the individual who was born before this increase, right? So it doesn't seem like there's much turnover in the human brain. And to support this study, there's another post-mortem study, human brain study. They checked the brain sample from the patient who received this reagent called BRDU, bromodioxyurea, which also will be incorporated into a DNA when cells are dividing. So you can only see dividing cells. And this method is so much more sensitive than comparing the level of C14. And you can see occasionally BLDU positive nuclei in these patients. However, none of them colocalize with neuronal markers such as neuron or neurofilaments. Only time they see was colocalized with GFAP, which is a protein present only in astrocytes. So what this study tell you is that neurons are not really generated in adult human brain. So, although we are going to learn about repair in nervous system today, it seems like repairing nervous system by replacing with new neurons doesn't seem to be our option, unfortunately. So the good news is the processes, axons and the dendrites, can be regenerated once they are damaged. Although even that is limited to the peripheral nervous system. And when it comes to the central nervous system, it's very difficult for them to regenerate their axons and dendrites. So, the reason that we know the peripheral nervous system can regenerate the axon is because it was revealed by this courageous experiment done by this person, Sir Henry Head, an English doctor, over 100 years ago. He decided to do experiment on his own arm. Would you do that? No. So he made 2 cuts onto his external cutaneous nerve, the nerve that sends sense of skin. And as a result, he lost sense of a large part of his left forearm, which is marked in the top panel B. Then he decided to monitor how his sense of skin came back. Yeah, it took months for his sense. At first, very extreme sense, such as pain or extreme temperature, very hot, very cold, came back. But for more subtle sense, such as light touch or moderate temperature, it took so much longer. But eventually, he regained his sense of skin. And then he made a test by applying 2 touches. And they measured the distance to see how far they have to be. So he could tell there are 2 touches. And those positions were marked in this lower panel, B. By the way, this test is still used these days to evaluate the sensitivity of the skin. So what was happening in his left forearm? So when the axons of these neurons are cut, it causes inflammation. And as a result, a cell type called macrophage will respond to it. What macrophages do is to clear out debris. So there will be some Schwann cell debris in addition to axonal debris. So macrophages will chew on them and clear them out. In the meanwhile, neurons will detect that their axons were damaged. And that would trigger expression of a series of growth-related genes. And as a result, there will be a structure called a growth cone appears at the tip of their axon. Do you remember what the growth cone is? Anybody? Growth cone, you can see at the tip of axon of young developing. And it has a hand-like structure and it's very mobile. And it seeks for cues. There are a lot of cues. Some of them are attractive, some of them are repellent. and makes decision on which way to go. So this growth cone will reappear in the adult axonal tip and then starts growing back. In the meanwhile, because some of the Schwann cells are also damaged, that would trigger proliferation of Schwann cells. And those newly sensitized Schwann cells will start wrapping up regenerating axon with myelin sheath. So here's more realistic drawing of what's going on. So if you look at the left panel, this brown thing is the axon, and the green thing is the Schwann cell. The Schwann cell will be wrapping up the axon with myelin sheath. And the cut is made over here. And what happens is it triggers inflammation, and then macrophages will respond to it. And if you look at the middle panel, you see macrophages chewing on debris, Schwann cell debris and axonal debris. And then what happens is, if you look at the next panel, at the tip of the axon, you can see growth cone appearing, and then starts growing back. If you look at the right side, there's a picture of the real neurons, real axons, and you can see a lot of growth cones are appearing at the tip of their axons, right? And if you look at the lower part of this picture, you can sort of see where the old axons are running through. These axons will be cleared out eventually by macrophages, but the surrounding environment, such as extracellular matrix, will be still there. And that would help a lot for regenerating axon to know which way to grow. So they can just trace back where they used to run. So let's look at the real axon regeneration. I will show you some footage. So did you notice that the speed of the axonal growth is not constant? Did you notice that? At first it was kind of slow, and then in the middle there was a moment it was growing very fast, and then it got slowed down again. Can I show you again? It's kind of slow in the beginning. Now it's very fast. And then let's slow down again. So what this study was showing is that when they are slowing down, that's when they encounter a lot of garbage, debris. It correlates when they are going through debris excess, right? So it's very important for macrophages to get rid of the debris very quickly. That promotes the axonal growth. Another factor that regulates the speed of the regrowth is that neurotrophin. So if the neurotrophin is released from target cell, that would promote axonal growth. So if the animal goes through a lot of exercise, and then the muscle will release a lot of neurotrophins, and that will promote axonal growth. So if, in this case it's mouse, if the mouse goes through rigorous exercise, treadmill 10 meters per minute, it's very fast for us. And 1 hour per day, 5 days a week. And in 2 weeks, you can see as compared to unexercised control, there's tremendous difference in axonal regeneration. So, as we looked at so far, peripheral nervous system can regenerate their axon, and then they can trace back where they used to run. But in the end, it has to make connection with the target cells. How do they know where to terminate? Well, if you look at the synaptic site, as we can see in the top panel, Schwann cell still there. And if you look at the right panel, acetylcholine receptors, that means postsynaptic site is still there. So those structure remains. And that would help a lot for regenerating axon to know where to terminate. So the lower panel shows before and after the injury. So before the injury, this axon from motor neuron is marked in green, is making connection with 6 different muscle fibers, which is highlighted in red. Now, 25 days after the injury, this regenerated axon is actually making connection with same set of 6 muscle fibers. So, the peripheral nervous system can regenerate their axon, they can run, they can trace back where it used to run, and then make connection with the same set of target cells. However, as I said earlier, central nervous system has a lot of trouble doing this. And actually, we know that for a long time. It's even described in this ancient Egyptian papyrus document. I don't know where exactly it says so. But according to your textbook, somewhere in this document, it says it's very difficult to repair brain and spinal cord. So what's happening if the CNS neurons are injured? Well, if the axons of these neurons are damaged, that would cause inflammation. And then a cell type called microglia will respond to it. What microglias do is to clear out debris. However, it doesn't do as good job as macrophages. It's kind of slow. So you would see there's a lot of debris. So in the CNS, oligodendrocytes will make myelin C, not Schwann cell, oligodendrocytes. And there will be a lot of myelin debris. And as we looked at earlier, we know that if there are a lot of debris, that slows down the regrowth of the axon, right? So it's bad news. But to make it worse, is microglia cells attack intact neurons. Not only the injured parts, but also chewing on intact, not injured neurons. So it makes the injury even worse. And to make it even further worse, there is another non-neuronal cell type, astrocytes, will also respond to inflammation. And it changes morphology and becomes highly proliferative. They are called reactive astrocytes. And this reactive astrocyte will release a lot of inhibitory factors for axonal regeneration. So neurons are not quite encouraged to regrow. Instead, they were pretty discouraged. So one of the well-known inhibitory factors is called NOGO. Do you know why it's called NOGO? It says no go to yes. So, whether the most common injury that's done in the CNS is caused by lack of oxygen, called hypoxia, or lack of blood supply, ischemia. Why? Because brain is very energy demanding piece of tissue. So it requires lots of oxygen and glucose. And if you lack the supply for a minute, they can't tolerate. So it will make injury. So in the left panel, it shows the example of damaged brain. This is visualized with a technique called Niesel stain. Niesel stain, you can stain soma of neurons, and you can see there's huge damage in the right hemisphere. And what's happening here is visualized using another technique called immunostaining, and showing on the right panel. So immunostaining, this technique uses antibody against specific protein. In this case, protein which is only present in neuron, and then stain with fluorescent dye, which is red. But in the meanwhile, you take another antibody, recognize only protein present only in dying cell. And then you take another fluorescence, green, and then stain with it. So if the cell contains both of the protein, and then it will be both green and red. So as a result, you see yellow. And you can see a lot of yellow cells in this picture. They are dying neurons. So that's what happens. So what's happening in these neurons when they go through hypoxia or ischemia? As I said, the brain is energy demanding tissue. So if you lack oxygen or glucose, and then you can't sustain membrane potential. So neurons will get depolarized. And what happens when you get depolarized is releasing glutamate, right? So there will be a lot of glutamate release. And glutamate will activate glutamate receptors, including NMDA receptors. Do you remember NMDA receptor? It's a ligand-gated ion channel, right? What ion? Remember? Calcium. Yes, it's a calcium ion channel. And I remember telling you that NMDA receptor is kind of tricky ion channel because it's a ligand-gated channel, but glutamate binding itself is not enough to open it. It has to be depolarized. So usually NMDA receptor is not easy to open. But in this scenario, neurons are already depolarized. So a lot of NMDA receptors will open, and there will be a lot of calcium influx in the cell. Now, calcium ion is used for signaling. It's very important for the cell. But if you have too much of it, it's actually toxic to the cell because it attacks mitochondria. What's the function of mitochondria? Anybody? Yes, produces energy. So it produces a lot of ATP. And it's important for especially neuron because neuron is very energy demanding cell type. So it's important to have mitochondrial function because it's efficiently generate ATP. However, it's kind of like nuclear power plant. Once something goes wrong, it's utterly disastrous. And usually, it's protected by this protein BCL2. However, if there is a lot of calcium influx, and then this protein can no longer protect mitochondria. And then stuff leaks out from mitochondria, including cytochrome C. And once this cytochrome C comes out from mitochondria, it activates protease called caspase 9. And then that would trigger a series of activation of caspases. So we call it cascade of caspase activation. And as a result, caspase 3 will be activated. What this guy does is to go to the nucleus and then start chopping up proteins which are important for chromatin stability. So the DNA will become kind of loose. And then, in the end, DNA will be chopped up into pieces. So it's a very special way of death called apoptosis. So that's what's happening in these neurons. So this type of cell death is caused by a lot of glutamate release. It's called excitotoxicity because if the neurons are too excited and then releasing too much glutamate, that will be toxic. And in the meanwhile, because it was causing some inflammation, so the inflammation signaling cytokines will do more harm to the neurons too, because it also attacks BCL2. So they are destined to die. And what happens to the portion of the brain damaged? Neurons are dying. What happens to the space? It will be filled with non-neuronal cell types, such as astrocytes, oligodendrocytes, and microglia cells. And those non-neuronal cell types are present in a healthy brain. However, once the inflammation happens, that triggers proliferation of those cells, and then it will fill up the space like this. So it will basically make scar tissue. So that will make it even more difficult for neurons to regrow because it literally acts as a physical barrier. So, as we looked at, central nervous system is very difficult to regenerate. But that's because they are not given an encouraging environment, right? But it has a potential to regenerate their axons. And that was shown by this very elegant experiment. So the optic nerve, which sends the light input from eye, belongs to the CNS. So once they are cut, they can't regenerate. On the contrary, this sciatic nerve, which is sitting right next to it, and it's important for controls eye movement, these neurons belongs to peripheral nervous system. So it has the right environment, such as Schwann cells. So they decided to take some tissue of sciatic nerve and then graft it to optic nerve. And then they can regenerate the axon, and they can even make connection again, which is shown in here. This is the electron microgram. And the axonal terminal of optic nerve is highlighted in this blue. And if you look at inside of this, you can see a lot of small dots. Those small dots around here. Can you see? What are those? Anybody? They are synaptic vesicles. So that's presynaptic terminal. And if you look at the adjacent cells, where the arrow is pointing, you see some very dense dark area, right? What are those? Postsynaptic density. So that's postsynaptic site. So presynaptic and postsynaptic, they are synaptic sites. So far, we've looked at that the peripheral nervous system can regenerate their axons, and they can go through where you used to run and then make same connection with the target cells. But the CNS neurons cannot do it. And that's because they are not given the right environment. They do have a potential to regrow. So basically, the neurons in your head right now are the ones you get, and you're not going to get anymore. And if you lose some, that's it. And well, we live long life these days, so over the year, we might have a lot of cell damage and lose, and that might eventually cause problems, such as some neurodegenerative disease, such as Alzheimer's disease, which is a most common neurodegenerative disease. So this disease was first reported by Alois Astroff, Austrian doctor over 100 years ago. So he encountered this patient who was in mid-40s, but had a severe dementia. And then eventually, he had the opportunity to examine his postmortem brain. And then he found a striking abnormalities in his brain. And that was the beginning. So that was over 100 years ago. But these days, as our life expectancy become longer and longer, this disease is becoming a serious social problem. Right now, 10% of people over 65 are suffering this disease. And if you look at the population over 85 years old, 45% of them are suffering from this disease. So it's really a serious problem. So at first, in the early stage of this disease, patients will have retrograde amnesia. What this means is you cannot recall something that you used to. And then that will progress to anterograde amnesia, which means you cannot form new memories. So that's rather mild case of Alzheimer's disease, but it's a progressive neurodegenerative disease, and in the later stage, it will affect a lot of problems, a lot of brain functions such as language skill, problem solving, judgment. So it's very serious disease. So I would like to show you an example of patient who has still mild Alzheimer's disease. This is how it starts.
Okay, so I'm going to mention 3 words, and you say them after me when I'm done, okay?
Apple, penny, table.
Apple, penny, table.
Super, super, super. Can you do me a favor? Can you pull out your mathematics hat and subtract 7 from 100?
It was clear that she did not need a lengthy battery, that what we were into was someone in the moderate stages of dementia.
23.
Good.
Her memory is not going to be her friend.
Do you remember those 3 words I just asked you to remember?
No.
You remember any of them? No?
For example, she could not remember 3 items within 1 minute's time and could not do that over a period of time.
Did you remember all those 3 words? Well, you might have to watch out.
Just apple, right? Apple, I think juice.
Apple and penny. I don't remember the other one. We're all screwed.
But you want to remember at least one, right?
Yeah.
But she instantly said no. Not at all. She remembered. Well, that's some difference. Okay. So, in Alzheimer's disease, so you get a lot of neuronal death, but not all the neuronal type is equally affected in this disease. So some of the neurons are extremely sensitive to this disease. And one of the most vulnerable neurons in Alzheimer's disease is cholinergic neurons. So there are 2 cholinergic systems in the brain, which is highlighted in this purple and the green, and of which this purple one, basal forebrain cholinergic neurons, are the very first to die in this disease. It projects to all over the brain, so you can see a lot of cortex areas are innervated by this neuron. But one of the most important, I would say, projection is the one to hippocampus. And hippocampus is important for memory formation. So it sort of makes sense that in the early stage of this disease, you start to see memory deficit. So the abnormalities that Alois Alzheimer saw in his patient's brain were actually these 2 types of aggregates. One is called neurofibrillary tangles, and the other one is called senile plaques. And you would not see this type of thing in a healthy brain. It's very striking abnormalities. So what are they? How are they formed? It turned out that the plaques are made out of this short peptide called amyloid beta. Amyloid beta is actually released from neurons. And then it makes aggregates extracellular. And what it does is it impairs synaptic site. So the first thing happens is that they will start losing connections. And then those dendrites which lose a lot of connections, it's basically injury. So that would cause inflammation, and then microglia and the astrocyte, reactive astrocytes, will start making it even worse. And there is a substance called ApoE. This substance seems to help clearing out aggregates, the plaque. So if something goes wrong with this system, it also raises the risk of getting Alzheimer's disease. What about tangles? Well, it turned out tangles is made out of this protein called tau, which is in the cytosol. So tangles are, it's an aggregate, but it's formed intracellular. Plaques are extracellular. And these days, actually, we can monitor the amount of plaque in the live human brain. It used to be only available for postmortem pain, but we don't have to wait until the patient passes away. We can monitor, and it turned out some people have plaques and they don't have any memory deficit yet. It seems like at least 20 years before you see any symptoms, these plaques start to form. So it's a very long process, these diseases. Yes?
Sorry, so the alpha, beta, what was the name of the protein? Alpha, beta?
Amyloid beta.
Amyloid beta.
Amyloid beta, A, B, cells.
And they aggregate when they're released? And then they?
Yes.
Okay.
Yes, I will, yes. The question was, so what is this component makes plaque is called amyloid beta, and I will explain how it's formed in a minute. So, let's talk about tau first. So tau turned out to be a protein which binds to microtubule. And what it does is it stabilizes microtubule structure. And its function is regulated by phosphorylation. But in a pathological condition, tau gets hyperphosphorylated. And when it's hyperphosphorylated, it tends to make aggregates. So you get tangles.
Thank you.
So if that happens, and then there's no function in tau, so as a result, microtubule will be destabilized. Microtubule is a major cytoskeletal component in the processes, axons and dendrites. But it's not merely just a cytoskeletal component. It acts as a railroad. So neurons synthesize some membrane protein or secreting protein, and then it has to be delivered to the peripheral side. So it's a railroad to deliver that. And sometimes they want to bring something back to the soma from the peripheral side. So this trafficking will be impaired. And that's a very serious problem for neurons. So it's becoming unhealthy. Now, let me explain where the amyloid beta is coming from. So amyloid beta is actually turned out to be a part of this membrane protein called amyloid precursor protein. And this protein can be cut extracellularly, but if these two sets of enzyme protease called, one is called beta secretase or base, and another one is called gamma secretase or presenilin. So if this protein is cut with these two sets of proteases, and then this part of the peptide comes out, and this is highly, it has very high tendency to make aggregates. So there are other proteases too, and if it's cut in the middle, for example, and then that's fine. So it's just a matter of combination. And if these two cuts and release this part of the protein, and then that's a problem. And there are certain Alzheimer's patients who has familial history. So a lot of people in the same family get Alzheimer's disease. And these patients tend to get Alzheimer's disease much younger than average people. And we call it familial Alzheimer's disease. And scientists found mutations in these familial Alzheimer's disease patients' gene. And if you look at where the mutation is, APP, presenilin, base one. So it seems like these are the key players. To confirm this, scientists made transgenic mice carrying those mutant APP or presenilin, and those mice found the plaque. And their memory is impaired too. So they show Alzheimer-like symptoms. So they do get Alzheimer's disease. Mouse can get Alzheimer's disease if they carry this. So it looks like this formation of plaque is a key factor. But the puzzling thing is that only maybe 1% of Alzheimer's patients you could clearly explain why those people are getting Alzheimer's disease. Other people, you don't see a clear genetic basis. Vast majority of Alzheimer's patients, you can't really see clear reason why. So, to think about why, I want to introduce this case. So these days, it's becoming a big problem, right? Football players seem to get a lot of brain injury. And yes, it looks like repeated concussion will cause serious brain damage. So post-mortem human brain samples are shown here. Left one is from the NFL player who died at 45, very young. And as compared to normal post-mortem brain is 65. It's 20 years younger than that. And you can see profound damage was done in this brain. And if you look at in detail, you could see tangles in this brain. So it looks like something like repeated concussion, or it could be some very local ischemia or hypoxia could trigger some damage, and then that would form some Alzheimer's like aggregate. And then once that's going on, it's a chain reaction. It gets worse and worse over the year. So that could be the reason. So what do we do if we get Alzheimer's disease? Any cure? Well, there are several FDA-approved treatments for Alzheimer's disease. And you can categorize into two groups. One group is acting on acetylcholinesterase. It's an enzyme which degrades acetylcholine. So as I mentioned, cholinergic neurons are the most vulnerable neurons in the disease. So in an early stage, you start to see memory deficits. And if you inhibit this enzyme, and then you can slow down the degradation of acetylcholine, as a result, you can sort of boost the level of acetylcholine in the brain. And as a result, patients perform better in a memory test, for example. But obviously, this is not really solving the problem. It doesn't stop or slow down the disease itself, and it's a progressive disease, right? Another group of treatment is that NMDA receptor agonists. So as I mentioned earlier, the brain damage is done often by excitotoxicity. So if you block NMDA receptor, and then it might minimize the damage, the brain, the neuronal cell death in the brain. So that might be a little more promising, right? However, this drug, memantine, there's a study showing that it might not work as we have expected. So there's a lack of evidence of efficacy. So what do we do? Well, did any of you see this PBS Novel program on Alzheimer's disease, which was aired about 2 weeks ago, I think? So they were reporting this new human trial, and what they're doing is they decided to use antibody against plaque, and they inject the patient. So the idea is to use your immune system. And you have this antibody recognizing the target, and then your immune system will clear it out. So that's really tackling on the problem itself. So it sounds much more promising than this pre-existing treatment, right? So it's just the beginning of human trial, and so far it has very small but promising results. So if they give highest dose to the patient whose stage is a little bit moderate, not the advanced stage, and then you seem to have a little bit difference in the progression. But there is side effect too. Some people show swelling of the brain. So in any event, immune system, using your own immune system, and immune system is very complex and might be very different between individuals. And well, any clinical trial takes a very long time to get it approved. It takes more than a decade, and it's just started, but we'll see. It might be a very useful way to cure this disease. But for now, I mean, it might take decades to be available. So for now, is there any way to prevent or slow down the disease? So scientists using this transgenic mice carrying this familial Alzheimer's mutant genes, so it makes plaque. They found that those mice, if they are kept in an enriched environment, and then it can slow down the progression of this disease, plaque formation. So it's very important. Don't get bored. So let's just overlook what we learned today. So first we looked at how the peripheral nervous system can regenerate their axons and then both go through it used to run and then make same targets, and that's because they have an encouraging environment, such as Schwann cell starts wrapping up with myosin again, and also neurotrophin released from the target cell. But CNS neurons cannot do this. And that's because, not because they don't have ability to do so, it's because they don't have right environment. If they are given those environment, then they have the potential to regenerate their axons. And then we looked at age-related neurodegenerative disease, which could be very relevant to a lot of people. Okay, any questions? Yes?
Can you speak to any evidence of Alzheimer's being linked to exposure to prions?
Prions. Prion is actually kind of similar to AP. And I don't know if it's clinically, there's some connection to it, but prions is also made out of membrane protein, and then it's cleaved. And the way it's cleaved is actually the same proteins, gamma and the beta and the gamma sequence. So there's something we don't quite know, but nervous system, especially in the brain, is using those. I mean, they are not there to make trouble. They're using it for something else, and we don't quite know yet. And something goes wrong, obviously, and makes trouble. Yeah. Anything else? All right. So that's it. Okay.